Hello, Lokya. Am I audible? You are on mute, I think, Lokya. Lokya, can you hear me? Lokya, can you hear me now?
Yes, ma'am.
Again, there is some problem with other system.
Ma'am, actually, I came to my village, so the net was not working in the laptop, so I connected through my phone now.
Acha, okay. No. So, shall we start?
Yes, ma'am.
All right. So, today we will deal with two tools for primary structure analysis. So yesterday we have seen databases basics, how to download the FASTA, PDB, and classification of databases, how to navigate through the databases. Now that we have learned how to take FASTA file, we will see how to use this FASTA file in basic analysis. So first tool that is used for basic analysis is called as plot scale. It is very useful when you don't know anything about the protein and you just have to do the preliminary analysis. That is known as broad scale analysis. So, broad scale allows to first we will see what does it mean, and then we will see what is the algorithm of calculation of this, and then we will see hands-on exercise for that. Actually, most of the tools in bioinformatics, not only this, it everything happens at the back end. Tool is trained in that way to do the analysis. And so most of the times you just have to give your input either in the form of sequence or the structure, and you will be coming out with the output. But I feel as a good bioinformatician, you should know also what is the tool actually doing at the back end. For that, always teach algorithm and science behind it. So what is the plot scale? Here also you will see in the server, you just have to load your FASTA sequence and you will come with the analysis. Prot scale allows to compute and represent the profile produced by any amino acid scale on selected protein. And what is this scale? Scale is a value, numerical value assigned to each type of amino acid. Amino acid scale is defined by numerical value assigned to each type of amino acid. These values are based on the weight lab. Okay, so in the weight lab experimental data with the property, different amino acid scale values are calculated and they are reported as the scale values. Most commonly, this type of analysis is used to understand the characteristic of the protein, whether it is a receptor protein or it is a membrane protein or it is a soluble protein. So in that, the generally or frequently used scale is hydrophobicity scale. That means if a protein shows hydrophobicity at a larger ranges, that means all the membrane or receptor proteins are hydrophobic proteins. So that means you can say sure that the hydrophobicity scales, if they are or the peaks are at higher ranges, that means the protein which you have given as a query is a transmembrane or a receptor protein. So this data is made up of almost 50 predefined scales which are entered from the literature. And the technique is known as sliding window technique. Sliding window technique means at a time you have this window size in the ranges of odd numbers. So it can be 5, 7 or 9. Okay, so this window size is the what is mean by window? Actually, I'll tell you. At a time, how many amino acids at a time you are considering for putting or for drawing the plot is nothing but your window size. So if at a time I am was considering 5 amino acids, that means my window size is 5. If 7, so that means it is a 7. So window size is the length of the interval to use for the profile computation. That is number of amino acids examined at a time to determine a point of characters. So for example, hydrophobicity, how many amino acids you are considering at a time is your window size. Then why I'm calling it as a sliding window? So at a time you consider suppose first 5 amino acids, then you plot on the basis of amino acid scale values, you plot a 2D plot. Okay, you draw 2D plot. Then you move to the right by 1 amino acid. So it is like your window which is sliding. Shift to the right by 1 amino acid. So what happens? Next 5 amino acids will be considered. Then their values are put. So this complete 2D plot from first to last amino acid, it looks like an ECG kind of a diagram with peaks and troughs, crest and trough. And thereby you extrapolate these values on the x-axis, and this kind of plot is called as a Prot scale. Okay, so let's see. It runs an XPASI server. Here, yesterday we have learned either you can give uniprot number if you remember or accession number, or you can paste the sequence in this window. And there are properties, you can select any one property at a time. So here it is by default hydrophobicity, kite and Doolittle. You can select that. Okay. This kind of image you get, which is known as Prot scale output of a human. So now how to read this? If you extrapolate these peaks on the x-axis, that means there the particular characteristic is strong. For example, if I extrapolate this first peak on the x-axis, it is 10th residue. If I extrapolate here, then it is, if you can see, it is close to 40. So that means it is 38. The last peak, if you see, it is close to 100. So that means it is 98. And this is the fall. So here, that means it is the least value. So indirectly, what does it mean? The Residues at this position are hydrophilic and not hydrophobic. Okay, so this is the first simple exercise that we are supposed to do. Then after that, I will explain one more TMHMM and prot param. Let us first do this one by one. We will do. So we have to simply go to Google Chrome and type prot scale. Here you can either enter the Uniprot ID, accession number, or you can copy paste the sequence. So I'll copy paste the sequence. Let us do it quickly from Uniprot. Yesterday we have learned, so it will be quick for you today. Then we can go to submit button. Yeah. So the same thing which I showed to you in the presentation, only the thing is color, they have now changed it to golden. Same interpretation. What you are going to give me as a result will be this image, and below that you will write the positions of amino acids where you can see tallest three peaks. Okay, so the tallest three peaks are 1, 2, 3, 10th, 38 and 98. You can do it for any protein of your choice, or if you want to do it for same insulin, you can do it for insulin. Okay, Lokya?
Yes, ma'am.
Okay. So do it and give me the screenshot in the group, or here also in the meeting you can give me the result because you can't share the screen. Now, once I see your result, then I will explain the
Uh,
next.
Ma'am, after we have pasted the faster results, which one should I choose?
Pasta sequence you have pasted.
Yes, ma'am.
By default, it will be hydrophobicity, kite and Doolittle. Is the button blue in color?
Uh, hydropath, like
Hydropathy means hydrophobic.
okay,
So you can just click on submit. If it is
okay,
not by default selected, then you have to select it. If it is
okay.
selected already, you just
Yes,
click on the submit button.
okay. I'll send you the screenshot.
Yes, dear.
Ma'am, I've sent it in the chat.
Yeah, very good. You just have to also, for your reference, below that image, make one Word document, name it as exercise 1 for today, and give it a name prod scale analysis, and just type your result with the positions. Like for example, 10th, 38 and 98 positions are showing tallest peaks. So hydrophobicity is maximum at these residues. Okay, Lokya?
Ma'am, can you repeat?
I said with this image, you will write prod scale analysis, and below that you will write interpretation. Since top three peaks are observed at 10th, 38 and 98 positions, hydrophobicity is maximum at these locations. Okay?
Yes, ma'am.
Ha. Write it, then I will, within a minute, I will start the next tool. Okay, very good, Laukya. Now I will start with the next tool, TMHMM. Yeah, okay. I was just cross checking the document. Yeah, so I'm supposed to cover one more tool, okay? That also I will cover. I thought if time is left, I will also show you one more this thing, structure visualization tool. Let us see what is target of the syllabus that first we will complete, and we are still having time, then I will give you some extra information. Next tool is prot param. Okay, it is similar almost. It is also on the XPASI server. Prot param is for the parameters of the protein. Prot and param. Prot param is a tool which allows the computation of various physical and chemical parameters for a given protein stored in Swiss prot or Tremble TREMBL, or for user enter protein sequence. Anything as an input you can use. Yesterday we have seen Swiss prot is nothing but UniProt. TREMBL is also a part of UniProt. Now, which are the parameters that you get to know in the prot param? The computed parameters include molecular weight, theoretical pi, amino acid composition, atomic composition, extinction coefficient, estimated half-life, instability index, aliphatic index, and GRAVI, means grand average of hydropathicity. So these many properties at a time are predicted in one prot param output file. So this becomes very useful for understanding the properties of the protein. Okay. So what are these properties I have just mentioned? Because when you are doing it, you should know at least what does it mean and why it is calculated. So first property that we will see is half-life. Half-life of a protein is defined as the time it takes for the concentration of the radiolabile target protein to be reduced by 50% relative to the level at the beginning of the chase. 50% of the protein is what you say, it's reduced. So that is called as half-life. Next is isoelectric point. Isoelectric point is the pH of a solution at which net charge of the protein becomes zero. So pH equals to Pi and it is net charge of the protein is zero. So at isoelectric point, protein has no net charge because the positive and negative charges are equal. Theoretical Pi is based on the primary sequence. Then the next is extinction coefficient. Extinction coefficient is a measurement of how much light your biopharmaceutical absorbs. This value is dependent on the protein content of your sample. So if your sample is having a protein content and you give it for analysis, extinction coefficient is dependent on the light that it absorbs. Molecular weight, you know, it is the sum of atomic weight values of the atoms. Relative value of protein occupied by only aliphatic side chains is termed as aliphatic index. So if you want to comment on the protein's thermal stability, aliphatic index is important. And the last is grand average of hydropathicity index, which gives again same that we have seen just before, hydropathicity value or hydrophobicity value of a peptide. It's very simple, same operation. So let us go to the prot-param server. It also runs on the XPASI. So I simply type prot-param. I have already copied, so I will just paste the same sequence. And then you should click on this one, compute parameters. Oh, what happened? Your sequence should be at least 5 residues long. Acha, it does not take the FASTA sequence, so it gave me error. So what we will do, simply remove this header line, only keep the residues. Perfect. So these are the values. In the CSV format also you can save it. Yeah. So this you just have to read. Extinction coefficient is 17335. Half-life is 30 hours. Instability index 40.33, aliphatic index and gravity. Okay, all these you can report. You need not write any result. Theoretical Pi is also 5.22 and molecular weight is 1190, 980.21. So this is your prot param result. Total number of negatively charged residues also you get to know, total number of positively charged residues also you get to know, total number of atoms also. Okay, so all these things are bit important when you start the analysis of the protein. Prot param report is always asked when you are documenting a protein's information. So I don't think you just, I think CSV format is this. What you can, I think, do is simply copy this and paste it into one document, or else if you have understood, no need to give any result for prot param. It's very simple. Then I'll show you in the continuation next one also. So both you can perform one after another. Then we go for TMHMM. TMHMM is a membrane protein topology prediction method. So once you understand that your protein is a transmembrane protein, how to validate your information? There is one more tool for verification known as TMHMM. TMHMM is based on hidden Markov model. So hidden Markov model is a very famous statistical mathematical model. It has lot of applications. So since you are learning BTech course, it will be there somewhere in the bioinformatics course. Hidden Markov model is used everywhere, not only in bioinformatics. So we have our GPS in our vehicles, in car. That prediction is also based on hidden Markov model. Weather forecast, whether it will be cloudy tomorrow or rain will be there. All these forecasts are also based on HML. How? Basically, frame meaning of the mathematical model remains same. How it is implemented in different types of studies is variating. So the basic concept behind HMM is you have input layer, then you give this input layer to the mathematical model. It will be processed in the way that is trained, and then you get the output layer. So in between what is happening is not known, so it is called as hidden Markov model, and it is based on Markov chain. Okay, so no need to go into much complications. Just remember that TMHMM prediction is based on HMM. So we all have other bioinformatics tools also which are based on hidden Markov models, secondary structure prediction, database interpretation, all. Now, how to use this is again very simple, and I have purposely taken this exercise. You can take any protein of your choice. I have taken hydroxytryptamine receptor 2A because it looks beautiful. That is the reason. We go to TMHMM, take the sequence, and it will give you nice peaks, I will say helices information. So there are 7 transmembrane helices. Such kind of protein is known as serpentine protein. And of course, if you have so many transmembrane helices, it is a receptor protein. So let us have a look on the operation of TMHMM. So I go to TMHMM. Here, as I mentioned, you can use the same thing, insulin, but it doesn't give you very beautiful result as you get it in this. So I'm going to type a receptor protein, hydroxytryptamine receptor 2A. Enter. I come up with the table in which I will select Homo sapiens. So I have to do go and click the accession, download, FASTA canonical, download, copy.
Ma'am, it's not
Huh?
showing for me. This hydroxytryptamine.
Why? Have you written properly? What it is saying?
1 second, I'll check again.
Ha. Just see first. I will explain you how to read the result, then you perform. Okay, so you should see this kind of result. Purple color transmembrane helices. I don't want you to locate all the positions, just count the helices. You can see 1, 2, 3, 4, 5, 6, 7 transmembrane helices, and the golden color patch is outside. Then the purple color is transmembrane. Then again inside, then again transmembrane. Then again outside. So like that, the portion which is in blue color is inside, and portion which is in golden color is outside. Portion which is in purple color is transmembrane. So you simply take this image, and as a result you can write there are 7 transmembrane helices, so the protein is receptor. Now tell me, what is your problem?
Ma'am, can you tell the protein name?
I will put it in the chat box if you want.
Okay.
Hydroxy tryptamine receptor 2A. You can copy this and simply paste in the UniProt. Then in the table, select Homo sapiens and use that sequence.
Yes, ma'am, I got it.
Got it?
Yes.
Put the image once you get the result.
Yes.
Okay.
Yes, ma'am, I have sent.
Okay. Very good. Perfect. So Laukya, since you are the only participant and you are doing it quite quickly, actually you finished everything which was there in this short workshop. But but as we have time, and if you are interested, I feel like teaching you some simple tools. If you are interested, we can cover,
Yes.
which is not which is not even the part. So I like, I'm not that much rigid as to what I am supposed to deliver, that much only I'll deliver. Since you're doing it so nicely, I will give you 2, 3 things extra also. So now primary structure is done. You want to learn, right?
Yes, yes, ma'am.
Otherwise, if you say, ma'am, it is the syllabus is over for the workshop, we can stop here itself.
No, no,
No
no,
problem.
it's fine, ma'am. I want to learn.
Ha.
I don't think
So
we'll get this much practical thing in the college, so this is very
yeah,
good for me.
Arey Lauke, actually why they have broken, I don't know. 10 days workshop is so useful for people like you. So you get to learn 25 tools I teach. So many
Oh,
things. This was just like a trailer, very much beginning. So after that, generally in 10 days workshop, I will just give you overview because we have a time and you
yes,
have interest to learn, I feel. Otherwise, I don't speak much. I just see how the student is good. I can
yes.
see you are very sincere. So I just feel like telling you. So after this, generally the session 2 is structure prediction, secondary structure prediction. That I will teach you now because we have time left and it is very simple. Then I teach Swiss PDB viewer, which is comparison and superimposition of two proteins. This is secondary structure prediction. This is SPDBV, in which you get to know if two structures of the proteins are homologous. So this superimposition, RMSD analysis, everything is there. After that, strings to check protein-protein interactions. It is done on the, it has vast applications nowadays to check in a particular pathway how these proteins are interacting with each other. So it was my work in the PhD also. So this is a very simple tool actually, but it can be used for simple applications and it can be used or extrapolated or exploited for complicated applications also. So suppose if there is like I my PhD topic was hemolysin inhibitors in hemolysis. So if hemolysis is one event, which protein along with the main protein, other proteins are involved. So that particular study is known as network pharmacology. It is nowadays very much famous for research papers. So this network pharmacology, the network of the interaction is built by using string. Then I teach visualization tools. If now the time is there, I'll show you one demo of these visualization tools also. And the very famous rasmol, pymol. This is string. This is rasmol, in which you have few commands. Pymol also has graphical, both this rasmol, pymol, both of them have graphical user interface as well as command lines. So you learn simple commands in this. Then there is sequence alignment. This I'm sure you're going to learn. All this you're going to learn in your course. So I'm not sure how much practical they give you in your college, but they should include all these things ideally. Then blast, pairwise sequence alignment, primer designing, algorithms behind everything, how to operate blast, how to analyze, how to make primer reports, which is very important nowadays in the pharmaceutical companies also. Especially this also gained a lot of importance after COVID. How to make the primer reports and only design those primers which are essential or valuable in the market. Then I teach multiple sequence alignment, phylogenetic analysis, how to build the phylogenetic tree, how to inference about the evolution of any protein, any gene in the particular taxonomy. Then genome browsing by NCBI, UCSC, very important things of genome significant regions. Then Interpro, which is, yesterday I showed you this. The PFAM is a part of Interpro. So Interpro has 11 databases which you can study at a time. So how to study it in the Interpro. Then FIRE2, which is a structure prediction. In that I also teach mutation analysis like this. You get to know which are the regions of mutation analysis. You can see and learn this profile, sequence profile, mutation profile. And the last is allo sigma analysis, where there are 4 cases you study for this kind of images you get. And you get to know in which region your stability of the protein is more, where it is less. This is based on the energy values, per residue potential values. You get to know which area of the protein is stable in terms of energy after the allosteric sites are mutated or they are bound to any effector. So all this makes my 10 days program. Now, for the easiness, like some people can't give 10 days. So depending on their interest of the topics they enroll for the workshop, so it is divided. So next time, if anything is coming up, you can enroll. Or if any personal interest from your side, you can write a mail to me. I will give you guidance. But as a workshop, it will be in the break. So today's part was only the basic. As I told, you have covered everything. You have performed everything. But since we have time left, I will teach you one more thing. We will just see quickly how to perform secondary structure prediction. So I will not go into much details about the algorithm, which is mathematical. I'll just teach you how to operate the server because it is easy as well as very useful. So go to Chow Fastman server. Simply give, since we have already the sequence, I will simply give and paste it and click on predict. This kind of beautiful image you get for your secondary structure. So see, the red bars are helices, green bars are sheets, blue bars are turns, and yellow bars are coils. Then here, this information is also written in terms of symbols. HHH is helices, EEE is your turn sheets, TTT is your turns, and CC is your coil. So the last line with the color and symbol is your final prediction. And what does this star say? Every 10th residue is marked as a star symbol. 10. So this is for ease of calculation. If I ask you, Laukia, tell me what is the prediction between 10 to 20. You just count the and see the final region between 10 to 20. So majority are EE. That means the prediction is it is in sheet structure like that. At the bottom, they have also given the statistics, helices, turns, sheets, and percentage. So if you want, you can quickly try for any protein of your choice.
Ma'am, I did for insulin.
You did also. Okay, fine. So the same, na? So you got result like me?
Yes, ma'am, but a very short result.
Okay, okay. Huh. No problem. I think, okay. I copy pasted the same sequence and it was, I think, hydroxytryptamine. So for me,
Yes, ma'am. Yeah.
yeah. Okay. And but one more tool is there, but it is you have to install both of them, and it is not actually the part of workshop, but I will just tell you. Visualization tool, so that also you do, and then we will stop. Ras small, if you are a beginner in bioinformatics, for these tools, Rasmol, PyMol, Discovery, Chimera, for all these tools, I have a separate workshop of structure, prediction, and visualization. It just got over in the last week. When it comes again, you can join. In that, I teach every visualization tool in detail. It is for 3 days and 6 hours. Right now, I will just, for your interest and you are quickly doing everything, so I'm showing you. You can go to this download. Then your Rasmol will be downloaded. Since I taught you yesterday how to download the PDB file, I'm showing it to you. You should also know how to use it. So now we have to be, I told you yesterday, PDB file cannot be opened like that. It has to have one visualization software. So Rasmol is one visualization software. Once you do its installation, it will be opening like this, RASV. So I'll double click, open it. This is the platform. Then you can go to file, open, and any PDB file you can open over here. So suppose I'm taking this. This is how you visualize PDB file. Now in this you have different ways. So whenever you are working on any protein and you have to publish image, you can go for publication through rasmol. It has many good features. I will share one written document for you for the rasmol. When you get free time, or if you are interested, you can try different applications of rasmol. You can try their commands also. So display, this is wireframe. If you want to change it to backbone, you'll click on backbone. If you want to change it to sticks, you will change it to sticks. If you want to change it to ball and stick, you can change it. Colors, you can keep it chain wise. It will be different like that. This is normal GUI. If you want to operate commands, you have to write the commands. So suppose I say show space information. So this gives me information. This is histidine kinase protein. You're understanding, Laukya?
Yes, ma'am.
Yeah. Then you can see the sequence. So if I, the point of telling you this is when you are doing any basic protein analysis, you should know how to see it quickly. You can just go to rasmol and very simple to operate. You will get information, visualization of the protein in a very nice way. Show sequence will give you entire sequence of protein. Then suppose I want to give entire protein as one color. So you can give that command, color red, entire protein will be red. Then suppose I want to zoom it, zoom 45, zoom out. So it will become small. Again, I give zoom 100, it will be like this. Now, if I want to see hydrogen bonds in this, so you can type a command as H bonds on. You will get the number of hydrogen bonds and the dot. If you carefully observe here in this window, these dots are the locations of hydrogen bonds. So it is a very big protein, so you are not able to see it clearly, but still I think you can see here the dots are there.
Yes,
That
ma'am.
are your, yes, that are your hydrogen bonds. So like this, you can type simple commands. If I want to now undo this, so I will say H bonds off. Yeah, purposely I wrote this because if this rasmol is very simple commands, but they have to be accurate. Spelling mistake, capital small mistake, space, punctuation. If it is wrong, it will type like this, unrecognized command. So you have to give a space. Here I made a mistake. I did not give a space. If you type H bonds off, then it will be off like this. Okay. So this was additional thing that I felt like teaching you. If you want, you can try it now. And I think you will need a data of this. Nah? You will not remember. So what I'll do is on the WhatsApp group after our meeting is over, anyways I will give you the recording also, and for all the topics which we have discussed, I'll give you written notes of it. Okay, do it.
Yes,
It
ma'am.
will be, yeah, it will be useful for you. So
Yes, ma'am.
yes, okay. And any topic, if you want apart from this, you can enroll for the workshop. And now you are just a beginner. Afterwards, if you feel like doing advanced courses, I mostly take nowadays, being a senior, advanced courses. These are the basic things also that I teach, but mostly my workshops are for 10 days docking and simulation. When you learn all these things, you can take my help. When you are good in this, you can join advanced workshops also. Okay, Lokia?
Yes, ma'am.
Yeah, so I think that we can, if you want, you can do it in front of me now, or if you want, we can stop here for today.
Ma'am, I don't think I'll be able to download a software now. The net is not that stable
Ha
right now.
ha ha ha. Okay, so do it at your time whenever your net
Yes, ma'am. I'll
supports.
definitely try.
Okay, dear. And if you have anything, you can reach out to me for your if you need my guidance. I'm always approachable for good students and beginners. Okay.
Yes, ma'am. Ma'am, I wanted to ask, are there like any internships online for uh, related to this?
Yes, yes. I do take internships. So you can write to me, Laukya, your what are your requirements, what is your syllabus. I'll accordingly I will make a framework for you. I'll give you the charges and the durations, and then we can do the internships. Okay. We give
Yes.
internships also.
Ma'am, can you give me your mail ID or?
Yeah,
uh,
yeah. Can you note it down?
Yes, please.
Ha. So I will spell it, okay, if there is.
Yeah.
It's Sharvari, Sharvari,
Yes, ma'am. Sharviara.
VARI. Or
Okay.
else, wait, I will type it in the chat box.
Yes, ma'am.
So there are different levels of internships. You can just tell me, you can mail
Ma'am,
it.
are they online internships only
Yeah,
or?
yeah, all are
Oh,
online. We don't work offline only.
okay.
I stay in Mumbai, so we don't have, and
Oh, I
our
thought because the I thought the company was like Australia based company, so
yeah,
you're
yeah,
like from.
Australia based. So my boss is in Australia, but I operate in India, and we work online. So no
Oh,
problem. All are in online projects, online internship, everything is online. Hmm?
yes, ma'am.
So you can write it to me, and then I will give you all the different types of options of internship. Then you can connect to me accordingly.
Yes, ma'am. Thank you
Okay.
so much.
My pleasure, dear. It was nice teaching you. It was very small duration that we spent together, but you are a
Yeah.
sincere, but you are a sincere girl. I would like to connect to you later also for your help. Okay. Yeah.
Uh, Ma'am, I also have one doubt.
Huh?
I recently did a GMP certification.
Okay.
Uh, uh, like, is it useful, like in the this industry, the biotech
Good
industry?
manufacturing practices, you are saying.
Yes, yes, ma'am.
What was it actually? Contents?
It was the basics only. In the pharmaceutical industries, how do you the
Haha.
validation and the auditing and stuff like that?
No, my dear, it is not directly not related to bioinformatics actually.
Yes.
It is definitely a good thing that you did it somewhere in the future. If you are entering bioinformatics industry and you have additional knowledge of this, it will. But it is not directly somewhere connected to this computational biology.
Okay.
All right.
Yes, ma'am.
Okay, so I'll then end the meeting. We completed everything before in hand itself. We learned some extra things also. I would be happy if you download on your own the RAS small. I will give you all the notes for that so that it is helpful for you to refer.
Yes, ma'am. Yes,
Okay.
ma'am.
Okay, dear. Chalo. Bye bye.
Thank you, ma'am. Bye.
All the best. Yeah, bye.